Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. This lecture is intended for use by students who are enrolled in my courses at Del Mar College for my Human Anatomy and Physiology 1 course. If you stumbled across this video in YouTube land and you find it helpful, great. All of y'all hit like and subscribe, and it will continue to update you when I add new videos or edit these videos. These lectures, by the way, because they are intended for my students, they're going to be done in a way that I want my students to learn the information. So your instructor may go over it in greater detail, more detail, use different terminology. You need to learn the information the way that your instructor wants you to learn it. These lectures are for my students the way I want them to learn it early on. Later on, we'll get a little bit more technical and complex and add more detail. So, if you're in my class following along, we're on page 19. We're going to be studying nucleic acids, DNA and RNA. Now, I spent a good number of years of my life, probably 7 of them working in research labs, 5 to 7 years working in molecular genetics labs and doing protein biochemistry, or we did what's called in vitro translation. I know this stuff very, very well. The level of detail that you need to know at this point is not that great. So I'm going to present it in a fashion that we can get a basic understanding, and then over time, again, as the semesters go on, we'll continue to add detail and complexity and explain this a little more thoroughly and more accurately and precisely. But for now, just learn these basics until you get this down, then we'll make it a little bit more tighter. So now, we're going to talk about nucleic acids. We've talked about nucleic acids before when we talked about the 4 major organic compounds. And we said that nucleic acids store and transfer genetic information. That information that gets passed from one generation of cells to another, or from one generation of people to another. It is the instructions for all the proteins that are assembled inside of our cells. Now, when it comes to the nucleic acids, there's some definitions I want to get across first, and they're on the bottom of page 19 here. So let me make sure I do them in the order that they show up. One of the things that the first definition is the nuclear envelope. The nuclear envelope is the membrane around the nucleus. I talked about this before when we did the organelles, but we have a lipid bilayer that surrounds the nucleus, and then we have a second lipid bilayer around the nucleus. So it's a double membrane. And in some areas of that membrane, there are holes or openings. Those openings in the nuclear envelope are called nuclear pores. So the nuclear envelope is nothing more than the membrane that surrounds the nucleus. It's a double membrane with pores or openings in it. A nuclear pore is an opening in the nuclear envelope that allows substances to enter or exit the nucleus. And you should write those definitions down. Now, I want to talk about a couple of other things before I get too far here. DNA can come in different forms. DNA is a very long molecule that's made up of these other molecules called nucleotides. And if we look at it, there's 2 strands of nucleotides that are twisted around each other in what we call a double helix. A helix is a coil, and it's 2 strands that are coiled around each other, so the form that DNA very often takes is called a double helix. And what we're going to learn over time is these little lines in here are chemical bonds between these molecules called bases. Those bases really are somewhat of a written language. It's a chemical language that we could read, and it tells us put this amino acid first, put this amino acid second, put this amino acid third, or whatnot. For proteins, it's the instructions on how to assemble proteins. And in this area, there's a certain number of bases that might code for a specific protein, and we call this a gene. A gene is a series of bases on a piece of DNA that is the instructions for the order of amino acids in a protein. I'm going to write that out. It's a series of bases, or we could say nucleotides, but we're going to learn in a little bit what nucleotides are made of, and one of the major parts of the nucleotide is this nitrogenous base. But it's a series of bases or nucleotides on a chromosome, I'll explain what a chromosome is in a minute. That is the instructions for the order of amino acids in a protein. And if I had a separate series of bases here, maybe a smaller one, this would be gene number 2, this might be gene number 1 on my piece of DNA. And I might have a third gene down here, and each gene is the instructions for a separate protein. It tells you how to put the amino acids for those proteins. And there are some bases in between genes, and some of that used to be called junk DNA. It turns out it's not all junk, and we're still discovering and learning at an alarming and an incredible rate more and more information about our DNA, about our genome. But suffice it to say that if I have a long piece of DNA with lots of genes on it, many, many bases, we can call that piece of DNA a chromosome. And actually, the DNA can take on 2 shapes. So now that you have the definition of a gene down, I'm going to erase some of this, and I'm going to give you 2 other definitions, okay? When we use the definition for what we call chromatin, we can say that chromatin is loosely coiled DNA. That's kind of a very simplistic view of it, and that's a Mr. Long definition, but for now, that's all I want you to know about it. If I talk about a chromosome is a densely coiled piece of DNA. So listen. Each gene on a chromosome is the instructions for the order of amino acids in a protein. One chromosome can have hundreds of genes on it and be very, very long, and some of those genes are hundreds of bases long. Now, there are so many proteins inside of our cells that if we did all of the genes on one molecule, it would be one really, really, really long molecule of DNA, and it would be susceptible to breakage and other things. So, the genetic information in our cells that is encoded in the DNA is broken into smaller chunks. I call them volumes of DNA. That's my terminology for it. Almost like volumes of the encyclopedias. If you took all the information in the Encyclopedia Britannica or the Funk and Wagnall's Encyclopedia and you looked at it, if you put it into one book, it would be unmanageable. That book would be this wide. You'd have to set it on a shelf, take it off, set it on a table, climb on a ladder to open it. Pages would fall out. The binding would break. It's not manageable. But if we break it into many, many, many volumes, then each volume has certain information. If I want to read about Afghanistan or Alaska, I go to the A volume. If I want to read about Mozambique or, Memphis, Tennessee, I go to the M volume. So the information is highly organized and I know where to find the information. Well, that's kind of how DNA is arranged. DNA is broken into volumes, so to speak, and each volume is a chromosome. Within a chromosome, each piece of information is a gene. Our cells have 23 chromosomes, and then we have a second pair of those 23 chromosomes. So we actually have 46 chromosomes in the nucleus of our cells. That's 23 pairs of chromosomes. What I mean by that is, if you have 23 chromosomes that were given to you by your mother, you have the same 23 chromosomes given to you by your father, so that you have 2 sets of DNA. It's almost as if your parents did this. What if your mom bought you World Book Encyclopedia and your dad bought you Encyclopedia Britannica? It's sort of the same information, but they're not arranged exactly identically, because then there would be copyright infringement. It's the same information in the same order, but they might present it slightly differently. They chop up the sentences and rearrange it a little bit so that each one reads slightly differently. That's kind of what happens in our cells. Our cells have 2 volumes, or 2 sets of volumes of DNA, 2 sets of chromosomes. and the information might be slightly different from each parent. So now, when I talk about chromatin and chromosomes, if I stretch the DNA out like this would be in the form of chromatin. There are some proteins that I can start to wrap the DNA around called histones, and if I start to take this piece of DNA and I start to coil it up like this around all these proteins, it becomes very densely packed and compacted. So a chromosome is a unit of information, some densely coiled DNA that is all compact like this. And then when I need to read it, I have to uncoil sections of the DNA back into chromatin. It's the same piece of DNA. It's almost like this. I don't have one with me, but imagine if I took a cord, like a cable that you plug something in. If it's all unwound, it's a piece of chromatin. If I took the same cable and wrapped it around my hand so it was a densely packed ball like this, then that would be a chromosome. If I uncoil it, it's back to chromatin. If I coil it up, it's back to a chromosome. So that one piece of DNA can be changed from chromatin to chromosome depending on if we need to read it or not. It works kind of like this. Imagine if I had a series of pages like your note set, but instead of in the note set, they're being bound here, what if they were fan folded like computer paper? So in order to read it, you'd have to unfold everything, read it across the table. And then when you're done reading it and you don't need to use it, you just need to move it around, you can crunch it back up or condense it back into chromosomes so that you can easily transport it. So if you want to go to the library and study, you're not dragging pages 20 feet behind you getting damaged. So DNA changes form from chromatin to chromosome. When it's in the form of chromatin, we can read the bases, sort of. And when we don't need to read it and we're done, we can compact it down into dense bodies called chromosomes for easy storage. I hope that makes sense to you. There's one or two other definitions. There's the nucleoli, and you can look that up if you want. And we're going to move on to the next page, page 20. Okay. So now, when it comes to the nucleic acids, there's actually two different nucleic acids found in our cells, DNA and RNA. So I hope you got all of this. Double helix is the form of a piece of chromatin or chromosome. When it's uncoiled, it's chromatin. When it's densely coiled, we call it a chromosome. It's the same piece of DNA. Our cells have 46 chromosomes or 23 pairs of chromosomes. Okay, you got 2 of chromosome number 1, 2 of chromosome number 2, 2 of chromosome number 3. The reason we have that, by the way, is in case a gene gets damaged, maybe you get a bad copy from mom, you should have a good copy from dad so that you can still make that protein necessary for life. If both copies are bad, then you may have problems. Anyway, so now let's look at the structure of DNA, and then we'll move on later on to another video to do DNA replication and protein synthesis. In order to understand those, we need to understand how DNA and RNA are put together. So in your note set, there's a little table that has DNA and RNA next to each other, and they share some common characteristics. Both DNA and RNA have these characteristics that we can talk about. We need to know first of all, we need to know this. All nucleotides are made of 3 things. The nucleotides found in our nucleic acids are made out of 3 things. One of them is called a 5-carbon sugar. Now, when we studied sugars for energy like glucose, we said those were 6-carbon sugars. 5-carbon sugars are used for structural things, and there are different 5-carbon sugars that can be found in DNA and RNA. A second thing that a nucleotide is made out of is a phosphate group, or a phosphate molecule. And we talked about this before. It's a PO4 minus, and there's some chemistry associated with that we're not going to get into. And the last thing that is required to form a nucleotide is what we call a nitrogenous base. Nitrogenous because they contain an atom or they contain nitrogen, and a base because they're basic in their pH. They would remove hydrogen ions when placed in solution, if you recall your pH lecture. So since all nucleotides are made of these three things, then deoxyribonucleic acid and ribonucleic acid, and their names are spelled out in the note set. You should be able to spell out deoxyribonucleic, all one word, and then acid, or ribonucleic acid. This one has one less oxygen than that one, and so they call it deoxyribonucleic acid. So now, if I know that all nucleotides are made up of these three things, the difference between DNA and RNA is which of those are found here. So, when it comes to DNA, the 5-carbon sugar found in DNA is called deoxyribose. And in RNA, it's called ribose. Now here's a little hint or clue. Anytime you have "-ose" at the end of word in biochemistry, it's a sugar. Glucose, fructose, ribose, deoxyribose, galactose, lactose, mannose. Anytime it has "-ose" at the end of the word, it usually means it's a sugar in biochemistry. So ribose and deoxyribose are two different sugars because one of them is lacking in oxygen. It has a different chemical formula. The second thing is the phosphate group. And if we look at the phosphate group, it's PO4 with a negative charge for each one. And I will usually abbreviate this as a little P with a circle around it to mean my phosphate group when we build these molecules. The last thing I need is a nitrogenous base. And when it comes to the nitrogenous bases, there are 4 different nitrogenous bases in DNA. One is called adenine, and it's abbreviated with the letter A. One's called thymine. It's abbreviated with the letter T. Another one is called cytosine, abbreviated with the letter C. And the last one is called guanine, which is abbreviated with the letter G. These are the 4 different nitrogenous bases that can be found in one of the 4 different nucleotides that makes up a molecule of DNA. In RNA, we also have 4 different nitrogenous bases. We have the same adenine, we have the same cytosine, and we have the same guanine. They are the same 3 bases. The only difference is in RNA, we have a stuff called uracil, which is abbreviated with the letter U. There is no thymine in DNA, and there's no uracil in RNA. There's no thymine in RNA, no uracil in DNA. Okay? So, if I wanted to build a molecule of DNA, we know that nucleic acids are polymers of nucleotides, long chains of nucleotides. So first I would have to assemble a nucleotide. I need to take deoxyribose, a phosphate group, and any one of these 4 bases. And so the way that they are put together goes something like this. I'm going to erase part of this table, and then we'll do this again with the molecule of RNA, but I just want you to be able to see this. Now, if I take a sugar, and I take a phosphate here, they're stuck together in a chemical bond, and it turns out that the sugar can bind to one of the 4 nitrogenous bases also. And this one's going to be thymine. So I already know that I'm dealing with DNA because I see thymine and not uracil. If I took another sugar and another phosphate and another base and another sugar and another phosphate and another base, and let's just do a whole bunch of these. I'm going to do one more. So these are the sugars and these are the phosphates. Because I see thymines here, I know I must be dealing with DNA. I know the sugar has to be deoxyribose. It's just the way that it is. It turns out that the sugar and the phosphate can form another bond with each other like this. And when these link up in this fashion, I have the sugar and phosphate backbone with a series of nitrogenous bases here. The order would be TGCCTAG for this particular group. Now it turns out that DNA is usually double-stranded. And I don't like that our book talks about this because there are single-stranded DNA molecules, there are single-stranded DNA viruses and things, but it's usually double-stranded. I only have a single strand of DNA here. Now there's a thing called the base pairing rule. Erwin Schargaff is a scientist that figured this out and called it the base pairing rule. And what he realized is that the physical structure of adenine, thymine, guanine, and cytosine are such that adenine will bind to another base, but it will only bind to thymine. Adenine will not bind to another adenine, it will not bind to cytosine or guanine. It will only bind to another thymine. And guanine will only bind to cytosine and vice versa. So the little saying that is often taught in courses dealing with this stuff is at the golf course. A binds to T, G binds to C, and vice versa. If I know that adenine can only bind to thymine and vice versa, if I try to add another group here, let's say I brought in a sugar and a phosphate, I brought in another nucleotide. The only one that will form a chemical bond here is going to be adenine and thymine. And then the only one that will bind here would be cytosine. The only one that would form a chemical bond here would be guanine and adenine and thymine and cytosine. And I'm going to get these chemical bonds between here. Turns out there's 2 bonds between certain bases and 3 bonds between others. I'm not going to get into all of that. And then we could add the sugar and phosphate backbone like this. Or sometimes scientists, to simplify, once I see that there's thymines here, I know it's DNA, rather than draw all of that, they simply draw the molecule this way. ACG, GATC, and then the other strand would be drawn like this. TGCC, TAG. Okay? And then we could put the chemical bonds in here. Adenine and thymine have 2 bonds, guanine and cytosine have 3 bonds, but we don't care about that. So now I have a double-stranded DNA molecule that looks like a little ladder. If I grab the 2 ends of the molecule and turn them in opposite directions, then those bases would start to get twisted around each other like this. And as I coil it up, the molecule will start to shorten, and the chemical bonds of the bases would be in between, or inside our double helix, and the sugar-phosphate backbones are wrapped around each other like this. So that covers DNA. We know the base, we know the sugar found in DNA is always deoxyribose. We know that phosphate is phosphate. It's the same phosphate in ATP. And we have adenine, thymine, cytosine, and guanine. All nucleotides are made up of these 3 things combined. And since DNA is a polynucleotide, if I string them all together in chemical bonds, I have a single-stranded DNA molecule. Because we know that the physics of the universe and the chemistry of these molecules is such that only adenine and thymine will bind to each other, and only guanine and cytosine will bind to each other, if I know the order of bases on one strand of DNA, I know the order of bases on the complementary strand. And we use this term over here, complementary, with an E. Complementary with an I would be like, oh, your hair looks nice today, or that shirt looks great on you. Complementary with an E means they complement each other and go well together. So, one of the famous questions, some of the questions we ask when we talk about DNA is, which of the following bases would be found in DNA? Which of the following bases would be found in RNA? Or the sugar found in the sugar-phosphate backbone on DNA is blank. The sugar found on RNA is blank. Or here's a famous question. I'm going to erase this. But this is how this tends to get asked. Below is a single-stranded DNA molecule. Please give the correct order of bases that would be found in the complementary DNA strand. And if I listed a whole bunch of bases here, ATA, CG, ACC, TAGC, I don't know, I just come up with some random order. And I said, this is the order of bases in a single strand of DNA. what would be the correct order of bases in the complementary strand? You just use the base pairing rule, and your answer would be TAT, GCT, GGA, TCG. We use the base pairing rule. And you would look for answer choice C or whichever answer choice, and you would choose that one as having the correct complementary strand. Okay? Now, one of the things we know about RNA is that RNA is usually single-stranded. And again, I don't like saying that because RNA can be double-stranded, especially tRNA. Okay? But when it comes to RNA, usually, according to our book, it's single-stranded. And so if I were to give you a series of bases here, the sugar and phosphate backbone like this, and all I have to do is on each sugar stick a base here. You could know right away that this is an RNA molecule because you see uracil. Therefore, this sugar must be ribose. The phosphate never changes, okay? So now, we've looked at the structure of DNA, we've looked at the structure of RNA. In the next lecture, I'm going to go over what we call DNA replication, and we're going to do protein synthesis, transcription, and translation, so that we can understand how our cells divide and how they build proteins. And there's a lot of medicine being targeted at the genetics and at the DNA and the RNA, and we're, in the future, nurses and healthcare professionals are going to need to know a little bit more about this stuff. So I'm just setting a basic foundation, a very simplified one, but nonetheless, we're setting a foundation so that later on we can add detail. So I hope you learned something. I hope you had as much fun as I did. And please do this stuff until you can't stand it. Do it till you understand it, and then do it 5 more times so that you can explain it to a classroom full of 7th graders and they know exactly what you're talking about. If you can teach this stuff to someone else, you will be prepared for the exam to make an A. You will know it backwards and forwards, okay? Thanks for watching. See you in the next video.